come to that point that the dialogue from border film, hum hi hum hai to kya hum hai, tum hi tum ho to kya tum ho. We have to be together in all the disciplines. Otherwise, if we start to become plant pathologists, soil scientists, entomologists, no, we are agriculturists. We have to produce something for the farmers which is good. And you will probably not like my second slide, but it's the reality. So in the first slide, you see Norman Borlaug said, the greatest ally of the pathogen is our short memory. If we don't have a disease for some time, we forget that. And then what happens? We think we don't need to invest. We don't need to put money into that disease. And then I have added that generations of mankind enjoying outcomes of breeding for disease resistance are the culprits. They should remember what you folks all around the world, including, small to bigger country, have contributed to make the fields disease free. Second slide, please. So people who are not breeders may not like it, but it's an unfortunate story that we have to like that breeder is our center forward. He has, if we all start to make a goal, if pathologist says, I want to breed a variety, if serial chemist says, I want to breed a variety, if physiologist says, I want to breed a variety, no. Again, hum hi hum hai to kya hum hai, tum hi tum ho to kya tum ho. So we have to be together and choose a captain, and our captain is breeder. Next slide, please. Next slide, please. So this is the model. As you see the triangle, this is exactly inverting the disease triangle in our textbooks, in which we have environment, pathogen, and host. And I have chosen to invert it so that we can make the disease going under control. And this model was chosen when in 1970s, Australia had a rust epidemic in the southeastern parts. And they thought at that time Australia was living in a pretty much colonial system that every state was more like a country. And you have to have actually special permissions if you are surveying samples. So after all the bickering, people thought, no, we have to have some national way. And those guys thought that Northerns have done some, curse on them. But then they thought, we have to do something to have a national approach. And Sydney University started actually, we were celebrating, celebrating 100 years of research this year, but we have to postpone due to COVID. So 1920 or 1919, we started actually surveying rust samples by Professor Waterhouse. And then, वो कहते हैं ना पेडिग्री भी मैटर करती है. So sometimes I mention that I'm a pedigreed horse for breeding for rust resistance because I have Dr. L.M. Joshi from IARI in my pedigree. He was teacher of my teacher, Dr. S.S. Sokhi. And then my PhD guru, Dr. McIntosh, had Dr. Watson, who was student of Dr. Stegman, and Dr. L.M. Joshi and Watson just studied together under Stegman. So I feel very proud that I am a pedigreed plant geneticist or plant pathologist to do this, but without credentials, if I don't do it, if I don't publish it, if I don't produce it, then I will not be justifying my pedigree. So sometimes my pedigree makes me work harder and to justify the role of my ancestors. So basically, if you look at this diagram, you can see pathogen research, host research, germplasm, breeders, others means that we are all working together. And then you can see that on the under pathogen, that particular nitrogen tank, liquid nitrogen tank has 100 years of rust history. We have been collecting samples for 100 years and they are stored there. And we have, I can't even count on fingers, it's probably more than 6,000 single isolates. And they are pretty well, you can say, like DNA probes. You put few on a set of genotypes or set of varieties, you tend to know what genes you have. And then again, it comes from scientists sticking to their guns. If you look at the Australian rust research in Sydney, so in 1974 epidemic, when we got this national wheat rust control program, Professor Watson thought that we have to have host research, we have to have pathogen research, and we have to use both through germplasm and giving it to breeders. And that is the key thing which I think made the model for breeding for disease resistance, not only in rust, but in everything. If we get together, again, if we get together, we can do a lot. But in many countries, stripe rust is done in Calcutta, leaf rust is done in Mumbai, and stripe rust is done in Rajasthan, or Jobner, you can call it. But because then people cannot talk. We are doing these three rusts under one roof. And you can see some of your colored people over, or our colored people underneath. So some of them are my students actually, who are working, like if you see down the bottom, that's Mandeep Randhawa, who is running with the pipette, who is running the stem rust screening in Kenya. And Mamta Shetri is actually running now crown rot screening or genotyping in Queensland in Australia. And on the other side, you can see two of my people who, one is my current assistant. So putting it together that we all communicate on a daily basis and keep it as a family. And we have to treat it as a family and have to deliver the milestones because we are funded by the farmers. And farmers pay about $2 million per year to keep our program going. Next slide, please. So if you look at, in early 2000, we became Australian Cereal Rust Control Program, in which some of the informal collaborations which were happening in CIMMYT and University of Adelaide, CSIRO in Canberra, were brought together under one umbrella. We are still the biggest partner in that, and my program director is director of the whole thing, Dr. Robert Park. And in CIMMYT, if you look at in today's date, Dr. Ravi Singh, he's student from PBI with Macintosh. He was my senior. And Sridhar Bhavani, who's head of rust research there from Hyderabad, did PhD with me. And Dr. Randhawa, I mentioned already, And a young girl from Pakistan who did PhD with me, and Dr. Sandhu met her. She's joined as a molecular biologist probably in November. And head of the new wheat program is head of the wheat program in CIMET is a graduate of University of Sydney who did uh, actually PhD on crown rot. So you can say that although we are not a big wheat producing nation, we are actually number 5, I think, on the list. And we export a lot, don't grow. We do about probably something like 8 to 25 million tons, but majority of them is exported. So that this sort of, you can say, commitment which I'm talking about of the staff in starting from 1975, and now we have almost 20 years, 25, 45 years. There's about 8 key scientists, and everybody kept rolling on a 5 years grants. We did not choose to go to jump the gun to get a promotion from one place to the other. We stood by our guns to make the world rust free. And if you see that the cost benefit ratio of breeding for rust resistance has been cost, the benefit cost, I should say, 23 benefit and cost is 1. That was done long time ago, and I think it's actually much better than that now. Next slide, please. So that's what I said already. That's the next one. So now you can press all that. We cannot start any breeding program. Like, that's some of the problems I think which we face sometimes in countries in the Indian subcontinent, that we want every crop to be equal. No, we can't. We cannot. These are the few things that you can see: late blight famine, rice blast, southern corn blight, stem rust epidemic. They were the key drivers. They had the economic importance that we can actually really invest some money to breed for rust resistance. And I can tell you that CIMET had forgotten that stem rust is a disease anymore. But UG99 really brought them back to the square one. And this is where Australia was leading, because we never, probably majority of my time went into stem rust breeding in the beginning, which still, out of the 5 genes, probably 2 are still on stem rust breeding, because we don't want to lose that disease. We don't want to lose to that disease. And stripe, of course, is most common now. Yellow rust, you call it in some parts. And this is where actually I made my name, because I have probably discovered more than 50% of the genes. Next, please. So the main thing is I have kept this talk pretty much basic and simple, because once we research any area, we can go into the detail. There's endless of literature on fingertips these days. But we always think the area which I work with is more important than what Dr. Jakhad and Dr. Sandhu work on. No. Again, you have to be hum hi hum hai to kya hum hai, tum hi tum ho to kya tum ho. And the second thing is, खुद ही को कर बुलंद इतना कि हर तकदीर से पहले खुदा बंदे से ये पूछे बता तेरी रजा क्या है. So these are the few steps which are there, are not taken by one scientist. Identification, one person job. Understanding pathogen variation, other person. And then it gets mixed up amongst all of us. And eventually, we haven't done the breeding yet. This is what we have get the recipe cooking a good dal. or cooking a good sabji, but we haven't made it. And you can imagine that if we are not getting together to get that ration together, we will not cook a good dal, we will not breed a good variety. So, and I haven't put a, I have some serial chemistry background as well, but I thought let's go on disease resistance today. But if we look at all the traits, see, talking a plant pathologist talking to or geneticist talking to a serial chemist, physiologist should be a regular event. I can tell you one example here. A physiologist came to me and she got some material which was mine. I mean, I worked on it, not mine, you can say. And she had data, but she did not know how to even analyze it. And she was relying on a busy professor who may be like Dr. Jakhar and Dr. Sandhu, who are very busy, and they cannot spend time on analysis. They have to delegate. And once I told her, I said, don't worry, that person is very busy, I'll help you. Look at what we found was, we found mesophyll conductance in wheat. There's a gene on chromosome 2A. That was basically, and again, I thought I'm busy. I can, I cannot do it, but I can do the interpretation. And I put her on to another colleague of mine who is Indian as well, and Dr. Sandhu knows Dr. Bansal, Urmal Bansal. And she is good on analysis. And since I gave the reins to her from the molecular side, I never looked back. So this is where another point which I want to make is that develop, mentor your subordinates, mentor your juniors in a way that they become better than you. Only then you will be proud of it. If you want to keep them lesser than you, none of will develop because you don't have time and that person will be frustrated. Next slide, please. So this is something we sometimes have to go to basics. One of the things I thought when I had 70 or 80% of my time helping breeders on very routine stuff. No research at all, but only screening, screening, and screening, which my predecessor did. And I really thought that where should I actually work so that I can not have much competition? So the question came to my mind is that when Borlaug and co, Rajaram Borlaug, they put the genes in. Rajaram was also a student from the University of Sydney. And they have not put all the genes from the old wheats into new short stature varieties. So I went back to a collection which was collected in 1920s. And this is where I found about 15, 20 genes. And I'm still finding. Still, the journey is continuing. Seed size variation is there. Physiological variation is there. Adaptation variation is there. This is where I would say that I'm happy to collaborate with people. Where, I mean, I have a Spark grant with HHU. I'll be speaking with them at 7:30. That's a physiology guy is working with us. So never limit yourself. Never put yourself in a cage. Always be open. What you can learn from a physiologist or what physiologist can learn from a pathologist or a geneticist or a breeder or a serial chemist, you never know because your thinking gets really tunnel visioned. And once you talk to somebody, you open that tunnel and it makes you think laterally and work together even in the DNA age, when we have a population which varies for a lot of traits, we can discover a lot on the same. So this is the population I used. Next, please. Screening. You look at, I'm standing with some key people from, you can say on the, when I say India on the top, this is Nilgiri Hills. And we have collaboration with them from Watson's time in 19, you can say 50s. And Dr. Athwal, Dalwag Singh Athwal, who was IRI DDG, or sometimes he's called DS Athwal or Dalwag Singh only, who bred the variety Kalyan Sona, because his village name was Kalyanpur. He is also a PhD student from our institute and was probably wrote the best paper with Watson on pyramiding of genes without DNA markers. With DNA markers, we can do it now, but they did it in the non-DNA age. Watson and Singh, 1952. And I really admire their value. And you can see here Dr. Brahma and Dr. Sorry, I'm just slipping the name. Sivasamy is there, and the other one is Dr. Just missing his name. I probably had half a bottle of rum with him one night when I was there, and he didn't let me leave his room until I get fully drunk. Thank God I'm not drinking these days. And then you look at on the my right, maybe your right, Kenya. We are like the astronauts there. So we screen there for UG99. And then down there we have PVI, Kobiti, Ethiopia, India. I mean, any country, never think borders. Rust or diseases, they don't know the borders. They don't need passport. They can travel. They can travel in the wind. They can travel on your clothes. They can travel on your shoes. They can travel on the luggage. But be careful that we have to be careful that once we travel, we keep ourselves clean and let this international collaborations going so that we can actually beat these enemies. Next one, please. Okay, sometimes we talk about types of resistance. Majority of the time people say, Dr. Jack or Dr. Sandhu, please tell me when you want me to stop because I don't have to finish these slides because these stories are not finishing the slides. These stories are more like sharing the thoughts. Okay? So, all stage resistance. People say seedling resistance, major gene. I call it as an army. And although I usually have a photo of army, but I just put, sorry, Dr. Sandhu?
No problem. Please continue.
Okay, please let me know when you want me to stop. Because I just went into the historical mode a little bit, but as you said, asked me to. And if you look at zebras are on the ground, and the Air Force is a Qantas sign which I have put kangaroo, which is Australian airline, which is now we can both fail. COVID has failed us both, but if we are together, we will win. We cannot use one type. My 20 years or 25 years in Rust research, I had always had major fight with people who are saying we need only adult plant resistance. I said, forget about it, right? And it has failed quite a bit from cement germplasm in India because once you want a yield of 7 or 8 tons or 9 tons, you cannot really achieve it only with APRs, adult plant resistances. You have to have the muscle, you have to have the bone. So I call adult plant resistance as the bone, and then all stage resistance is the muscle. Muscle, You can regrow, but there are not many bones in the system at the moment. Next, please. That's just the variation. I doesn't have to be talked about too much. You can say that was among that Bartkin's collection, which I showed you. Next, please. So once you get that kind of, maybe go back once. I will just make one point. So if you look at up to 3 or 4, maybe some 5s, that's often major genes. And after you go 5 and over, 5 to 7, even 8, you have the minor genes or APR genes. So next, please. So now the question is, you have to map them. And you have to cross it. You can see the plan on the side. Then if you find monogenics or digenics, you have to get the gene 1, gene 2, and you can simply do simple marker systems, which I can leave it for the next talk probably when I do it, more on genetics. And on the side, so your one major gene is relatively easier to get the gene locations, but when you go to the QTLs or adult plant resistance, it takes much longer. And eventually what we want to give is a marker which you see as a little dot near 90, or on the other side, yellow and blue dots. They are SNPs, which are these days most popular markers. So basically, that's just a sort of thematic thing which I wanted to put there, is that although the slides looks very simple, but it can take you, these days probably you have cut down the cycle to probably 3 years, but it can take even 10 years. Next, please. So then the product comes something like that. You have a map, and this is the work which Mandeep Randhawa did when he was a PhD student under the Australia-India program. And he found this gene, which is now, I think, maybe ineffective. But you can see down the bottom, we have found so many genes. And you can see in the photo greens and the other one. So some of these genes look so clean. And you can all, but you have to put them together with others. Next, please. This is the example of when you actually have minor effect genes, or you can say adult plant resistance genes. And you can see the peaks. So one of them is on 1VL, which is YR29 group. The next one is on 2A, the third one is on 3VS. So they will not give you 100% protection. They will not look green. They will look maybe 50%, 60%, 40%. When you put them together, they can give you 10%. Go next, please. So this is where you can see that once you put them together, you can leave 1V, 2A, the last 3, and compare it to the first one. So that's what together they achieve. So last 3 categories are giving you individual effects, and the first category gives you the effect of all 3 together, which is less than 20%. So that's what is, once we are together, we achieve it better. If we are not together, we will just be sitting in the 50s and 60s. Next, please. So this is where, I'm not really a hardcore molecular biologist. I've done all the techniques, but I'm not a practicing molecular biologist, but I guide students in that area. And these are the genes, and plus there are a few more now, which we have cloned and shown. First one is actually the Perrinen et al. Was the first stem rust resistance gene cloned, and Perrinen comes from Coimbatore. And before he came to Australia, scholarship was offered, I told him, after 1 year, you will work with my colleague, Evans Laguda, to clone some genes. And he did. And he's still doing it. He's part of all these three. And you can say that I'm not, it's not, we should not actually leave everything. For example, sometimes some of my colleagues may think, or students may think, let's clone, let's start cloning, which is more, no. Even if you produce good material for cloning, then you get your name on the papers. You can make them to clone it quicker. So that's what I did. Next, please. We have done markers for many genes. This is only a short list because people can put these markers, these genes together in the F2 stage in breeding program, and then they can actually really put the gene combinations together so that if a risk comes in, it doesn't take out. So we can actually have durability of resistance achieved in our material early, because if we can combine traits like rust with markers early, we can use some markers for protein genes, we can use some markers for other maybe traits, cereal cyst nematode, which may be common in Rajasthan, and then we can actually find more money to test the quality of wheat which we are producing. Next, please. So the main thing is that when you develop any marker, validate it. I have found some papers published by really leading labs. They publish in a high profile journal, but they forget what the value of that paper is. And those markers don't even work on the line they have worked on. I don't know how they have done it, but you can see here, if you look at the first two rows, first is OS 27858, then Westonian, they have A and G, and G is for susceptible. And if you look at the whole table, it's all G's, which means we just have to convert the G to A. The marker is really robust, so you can use this marker. And if it gives you half A's and half G's, the marker is useless. Or if it gets close to 80%, it's useful, but you have to check the parents before you start to breed. Next, please. I think looking at the time, Dr. Jakhar, should I maybe conclude here or keep going?
Continue, sir.
Okay.
Continue.
So then after we have all this material, we have to create variability. Then we start to use these genes in backrows, and which involves hybridization and selection, which is real, then you have given the material to a cook who is the breeder. He has to cook it well. Next. So there's nothing new in these slides, which I'm just saying to you. Double haploid, probably not used as much in India, but some people are using it, I know. And genetic transformation is the one which I want to put on. One day, it will be accepted. At the moment, it's not, because at the moment, cloning of genes, it have no use except we develop good marker. This is the area which probably we will benefit more once we have people accepting the transformed crops. Next, please. Just press everything. And again, these are the methods which every breeder is using. And I just wanted to put it there is that, again, double haploids have come into it, and double haploids have actually halved the cycle of not half, probably brought it down about 4 years less. Say normally it used to take 10 to 11, and now it takes about, you can say, half the cycle of wheat breeding in Australia, because you're getting them within a year, and then you can multiply them, you can test it straight away, so you are saving 4 years using double haplords. And combination of above, because you cannot rely, never, as they say, never put all your money in one basket. You have to keep all the things going. Next, please. Next, please. Sorry, next slide, please. So this is something, again, Mandeep Randhawa did. You can see some PVWs, DVWs, and try to put all these genes together. And the main thing is that sometimes what people have done is, as you see in the green block, they have some genes as well. And we have some examples in Australia that people, when they were breeding, they lost what the original parent had and gained the other one by losing those, which you shouldn't, which we have to be careful that let's keep what our parents gave us and then gain what we have to add on. So then all those genes, which you can see on each side, they are the gain over, but the genes are already there. Next, please. So it's basically comes key messages. Can we do individually or country or regional basis? Can we believe in international? So these are all these things I have already sort of covered. But the biggest issue is material transfer agreements, MTAs, you see. And this is what is killing the total collaboration at the moment. And I hope one day common sense prevails that we can do it very quickly. And in my 25 years, I have given material to some breeders. Sometimes I just feel scared that I hope nobody sort of, gets it and says something. But now the system started that if I give any seed to anybody, I have to sign an MTA, and which is with I've signed sending back across material to 4 breeding companies. I did it in 4 months, but one company still is not signing after a year. So you can imagine it can take a lot of, because lawyers on each side can be very smart, but lawyers don't know that plant breeding is not a process like the general courts work. So I think we have to soften rules on MTAs. And interdisciplinary communication, join hands together with private and public partnership, find time to attend meetings, which is Zoom has made it very easy. Next, please. Okay, I think on the very first slide, I forgot to mention the photo was not of my grandfather, that was actually William Farrer. I just got into the border film too much. But on the first slide, if you see the bearded fellow, is William Farrer, who was the first Australian breeder, wheat breeder, was a surveyor who had TV and came to Australia for fresh weather from England and bred wheat, first wheat, and he actually realized that European germplasm will not work in Australia